Welcome to the structure of DNA. First, as a refresher, DNA stands for deoxyribonucleic acid. It is an organic molecule, and first, similar to how amino acids are monomers of the polymer protein, we need to establish the monomer of DNA, which is nucleotide. A nucleotide is composed of a phosphate group, a pentose sugar, known as deoxyribose, and a nitrogenous base, all covalently bonded. The phosphate and pentose sugar are known as the phosphate sugar backbone, as it never changes, whereas the nitrogenous base can be switched in and out. This unit repeats over and over again within DNA. Now, the four types of nitrogenous bases of DNA are adenine, thymine, cytosine, and guanine, known as A, T, C, and G for short. A and G are purines or double-ringed nitrogenous bases, while T and C are pyrimidines, which are single-ringed. A and T pair up and C and G pair up using hydrogen bonds. A and T use 2 hydrogen bonds, whereas C and G use 3 hydrogen bonds. Now that we established the monomer, let's put it all together. DNA is composed of 2 strands in the shape of a double helix. This means that the strands face opposite ends, known as being antiparallel. Now we indicate that DNA is antiparallel by using a notation called 5' and 3'. 5' is the, quote, top and 3' is the bottom. Notice how 5' and 3' are opposite, representing antiparallel. This is the molecule of DNA written out with its chemical structure. It stores and expresses hereditary information by the patterns of A and T and C and G within the molecules, which other enzymes come in to read to make proteins, similar to how a computer uses ones and zeros to impart instructions. Now, this is admittedly a very complex molecule, so we are going to learn how to draw a simplified 2D version similar to this together. And now, before we go on, please pause this video, gather a piece of paper and pencil to draw along as I instruct. Alright class, let's draw together a 4-nucleotide sequence of DNA in a 2D structure. So first, we're going to write up here, we're going to do a 2D structure of DNA, and it's going to be 4-nucleotide. And make sure too, guys, when you're doing this, to use a pencil, as it's a very complex molecule and we'll probably make some mistakes and interchange parts, so make sure you grab a pencil and a good eraser. So 2D structure of DNA, 4-nucleotide sequence. So first off, we are going to want to do our phosphate sugar backbone. Now, because this is a 4-nucleotide sequence, that means that there are 4 phosphates and 4 sugars on both sides. So instead of drawing out the molecule in its entirety, we're just going to use abbreviations. So for phosphate, we're going to use a circle with a P for phosphate group. So we're going to do first here, phosphate, and we're going to have it connect to a pentose sugar, which would be a pentagon. There we go. Have it connect here, label this S. And then we're going to repeat this 4 times. So we're going to put a P here, connect. There we go. S and S. Perfect. So here we go. 4 phosphates, 4 sugars. Okay. So next we want to label our 5' end and our 3' end. This is the top, so we're going to do 5', and this is the bottom here, 3'. And one thing to remember is on the 3' end, you always have to add a hydroxyl group, or OH, to the bottom. Okay. So this is our sugar phosphate backbone. Next, let's add in some nitrogenous bases. So I'm going to put some random letters here. If you guys would follow on, it'd be awesome. So I'm going to do another bond, and I'm going to put a box for the nitrogenous base, and we're going to do A first. Then we're going to do here C, and let's do another A here, and the last one we're going to do a G. Perfect. There we go. So this is 1/2 of the equation. As you know, it's a double helix and antiparallel. So what we're going to have to do is we're going to have to switch this paper around. So I'm going to flip the sheet of paper over. So as you can see, this now side is now upside down, and we're going to copy on the other side. Now for me, it's a little bit easier to kind of work backwards on the other sides to make sure that all lines up. So first I'm going to draw in boxes here for the nitrogenous bases. 4 boxes, 4 nucleotide sequence. Here and then here. And so what goes with G, C. What goes with A, T, C, G, A, T. Next, then we're going to have to do the pentose sugar and phosphate. So we're going to work backwards. So I'm going to go draw pentagons again. Pentagon 3, 4. So then we're going to connect. And so this one, class, notice that I drew it a little bit too high. So I'm going to go in here, I'm going to erase this and make sure that it's a little bit under your nitrogenous base so you can properly connect it. Perfect. So we're going to connect, 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 and connect. Perfect. And this is an S, 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 S. So then we got to do our phosphate. So we're going to do a P here, connect, second phosphate, third phosphate, fourth phosphate, 5', 3', and then with the hydroxyl group. Now we're going to flip this back over. This is our anti-parallel DNA molecule. Now there is one thing left that we forgot, and that is the hydrogen bonds. So now if you remember from the first half, A and T have 2 hydrogen bonds, and C and G use 3. And the way you delineate that is that you use dots. So we're going to do for A and T 2 sets of dots, C and G 3, 2, and then 3. And there you have it, the 2D structure of DNA, 4 nucleotide sequence.